we better get started. So as Chris said, I'm part of the fish health group. So a lot of what we use molecular biology for is detection of parasites, okay? And that's obviously involving the actual pathogen itself. But also we do a lot of work on using molecular biology to measure the gene expression. So what's happening in the host when they're confronted with certain pathogens, etc. We also do quite a bit of other work involving all different aspects of molecular biology, so metagenomic analysis for looking at diversity of different pathogens, whether they be within the host or the environment. But fundamentally, the bread and butter of the work we do revolves around PCR, and not just any PCR, but quantitative PCR. So that's pretty much what I want to get across to you today, some of the uses that quantitative PCR can be used for. So I didn't really speak to Chris beforehand, but I take it you've all been introduced to quantitative PCR, so real-time PCR. I know that you can't have got this far without being introduced to PCR. So fundamentally it's very similar. I'll touch on a few aspects as we go on. Okay. So in terms of detection, and first of all, be aware that I'm talking about detection here. I'm not talking about diagnosis of any disease. I'm purely talking about just the detection of our pathogen. Why would we want to use a molecular approach for that? Now, this doesn't have to be a two-way street because of the time business. So fundamentally, it's written up there. You're probably all aware that not all pathogens are culturable. Okay, so we can't always isolate a pathogen from the host or the environment and expect it to grow. They just don't all do that. Likewise, some of them are fastidious, they're very difficult to grow, or they're really slow growing. Some of these examples, for example, a lot of saprophytes and fungal agents, whilst you can culture them, they just take a very long time, and we don't always have the luxury of time if we're trying to detect and use detection for possible diagnosis, etc. And then another obvious one is highly infectious agents that are too dangerous to culture. So, for example, taking it a little bit away from aquatic health, but say we're in Sierra Leone and we're playing with Ebola, etc. Probably not really going to be one to culturing it. If we were going to, other than seeing someone bleeding out, we can actually use molecular techniques to figure out whether the agent is actually there or not. Okay. So in terms of molecular biology approaches, we have a few of them. I've only put a few on here because we could just have the whole lecture. This is being recorded, so I'm sure it'll be on my line. Likewise, I have to be careful what I say, keep the obscenities to a minimum. So we will be just touching on a few of these, but if we break them down into three kind of main areas, we have nucleic acid-based amplification on RNA and DNA. So here we're relying upon an ability to actually amplify the RNA or the DNA, so make lots and lots of copies of it so it's easier to deal with and detect, etc. Now that's different to nucleic acid hybridization. Okay, so whilst hybridization is part of the amplification process, when we're talking specifically about, my cursor isn't working here, the nucleic acid hybridization section, we're purely talking about does something bind, and then it's usually, instead of detecting it with multiple amplification cycles, we might be using a very sensitive reporter, or we might actually be amplifying the signal. Okay, So we're not actually making more of the hybridized pairing. We're just trying to detect it, and it's usually through an amplification of the signal as opposed to amplification of the product. So that's one kind of main difference there. And then we have some other antibody-based methods, and they're a little bit, that whilst they still fall under the branch of molecular, they're not really involving RNA or DNA, which is fundamentally what you would associate with molecular biology. Okay, so some of the advantages of PCR, and I'm sure you've been told before: sensitive, specific, and rapid. Goes without saying that if we had some of those traditional cultivation methods, etc., that on slow-growing pathogens, that they're not going to be rapid. Okay, the sensitivity, well, the hybridization part of the PCR amplification allows for exquisite sensitivity, and which we'll see a little bit later on in some of the case studies. And we also get the specificity, so we can differentiate what we're looking for. Okay, so if we're going to choose our PCR-based method, you've all done conventional PCR with just normal gel electrophoresis, and you've probably seen, hopefully, it worked well. But you hopefully were also introduced to some of the pitfalls that can happen. So when you were looking at your gel, maybe you had explained to you that it's possible that you can have aberrant bands, so other bands forming on your gel that aren't necessarily what you're looking for, the product. So we've then got what we're mainly going to talk about today is the real-time PCR or quantitative PCR. They're not really synonymous. There are other versions of quantitative PCR, but most people interchange them these days. And we'll introduce you to at least two of the major dye binding or methods. Now, I'll ask now, I suppose. Have you been introduced to cybergreen-based real-time and the hydrolysis probe? So, okay, good. So it won't be totally new to you. Just kind of give a refresher. Multiplex PCR, here we're obviously trying to detect more than one product in the one reaction. And then there's reverse transcription PCR, and this is what we do if we're actually trying to detect an RNA target. Okay. You obviously are aware that we can't amplify RNA, so what do? We convert it using an enzyme that usually comes from some viruses, and we convert that RNA into DNA, which we can handle. Can't get away without emphasising the critical factors for successful detection. It's fine having a perfect assay to amplify, perfect PCR, but that's kind of in the latter end of the stages. First of all, you've got to get that nucleic acid, and you probably did some extractions, but in your classes. But every extraction has its pros and cons. So fundamentally, we want to get the highest quality and the most amount of nucleic acid out of whatever we're trying to detect our agent in. Okay, so we commonly refer to those as different matrices. So that can be anything from the host, and then would be dealing with things like tissues and blood, etc., maybe mucus, swabs, etc., or the environment. We might be talking about water, soil, all those sorts of things. Okay, so when you are extracting the nucleic acid, you're not going to get 100%. It is lossy. You're going to have a very variable amount. So half of the successful detection is actually being able to get it out of your agent or your matrices, your matrix. Then you've got your DNA polymerase. A lot of people commonly refer to it as TAC. They're not all TACs, and the DNA polymerases are very different. Okay, some of them have very different functions, and these days DNA polymerases have evolved to the point where they've made a lot of them so are quite resistant to traditional inhibitors of the polymerase. So if we were trying to 10 years ago detect a pathogen in your blood. The blood would have so many inhibitors that would be co-extracted, so also be extracted with your nucleic acid, that it would cause lots of difficulties and basically reduce the sensitivity of the ability to detect whatever we're looking for in your blood using a particular polymerase. These days they've been kind of tweaked, engineered so that they're quite resistant. Okay. Then there's also some other little finer points on what they can cleave. You've got your choice of target, DNA or RNA. DNA is actually relatively stable. Okay. RNA, you probably heard, is very labile. Yes, it's labile if it is allowed to have RNases, the enzymes which destroy it, anywhere near it. And usually, within every cell, there are a lot of RNases. So these are the enzymes to break them down, and that's natural. That's to actually get rid of RNA and get rid of it quickly, because that's the messenger that goes through into ribosomes, etc., for production of proteins. However, having said that, RNA in theory is actually more stable than DNA if it's in its purest form and kept free of any particular enzymes or chemical situations that could cause hydrolysis. Alright, and then you've obviously got to be clean. We've talked about the exquisite sensitivity of these assays. No point if you're going to contaminate your samples. Right? So you've got to be very careful, especially if you're dealing with PCR. Why? Because you may have started off with a very small amount of what you're looking for, but then you've amplified it up into billions of copies. Okay, now we've done that once, and someone has opened up, say, one of your PCR tubes. Okay, there are billions of copies there. We were, say, looking for 10 before we amplified it. We're starting up another reaction. It's pretty easy to get some of those billion or more than 10 copies from that billion that you've opened up into another initial reaction. Okay, so contamination is always something you've got to be vigilant with. And you can get around that by obviously using dedicated work areas and equipment. And then you've got to have the appropriate controls. Okay, so you've got to be able to know if your signal that you're detecting is actually real. So you've got to have things like negative controls, positive controls. Otherwise, it's very hard to trust both positive and negative results. Are they truly positive, or are they false positive? Are they truly negative, or are they false negative? And usually it's the false negatives which are the concern, because they're quite easy to cause through things like inhibition of DNA polymerases, etc. Okay, so the first case study, and they're not going to be that in-depth. So this is a while ago now, so 2010, I developed an assay for detection of neoparamoeba purine. So this is, for all of you who know anything about fish, it's an amoeba that causes amoebic gill disease in the Atlantic salmon industry. First of all, it was a specific problem pretty much to Tasmania. It's now spread, and it's an issue through some of the really big farming areas or definitely through Ireland, Scotland. It's in Norway now. It's in Chile as well. So develop this assay to obviously detect it, and so that we can try and put in some management practices, etc. How did I develop the detection method? Well, originally it was based around cybergreen real-time PCRs, but we've since moved on and using hydrolysis or TACMAN-based probes here. To detect it, what are we looking for? We're looking for part of the 18S ribosomal RNA gene. Why do you think I'd be trying to look at that particular gene? That's the question. So if we're trying to detect a particular gene or some DNA from an organism, we probably want to look at a gene that has multiple copies. Okay, so in eukaryote eukaryotic organisms, the 18S ribosomal RNA gene is in multiple copies, and the beautiful thing is, in amoeba, it's in a lot of copies. Now I've calculated this at, let's say, approximately 3,000 copies for every cell. Now if we compare that to some bacteria, where you may only have a single copy, you can see that detecting one amoeba is the same as detecting, say, 3,000 of a single copy bacterium. So you're just increasing your ability, your chances of being able to detect it by selecting a gene that has lots of copies of what you're looking for. Okay, now this was just a very quick diagram figure I threw in this morning just to quickly reiterate what real time is. So if I point out and say this very first curve here, who knows the difference? Are there going to be more copies of, or more of our target in this sample to this one? Think back to your introduction to real-time or quantitative PCR. This is the cycle number along here, so PCR, so you know you went through a series of cycling, denaturing, annealing, extension, etc. And every single cycle, if everything works perfectly, you're getting a complete doubling of your product. Okay? Now with the real-time, all we do is have an ability to measure the amount of DNA after every cycle. Okay? And that's usually linked to a fluorescence, and we can detect that fluorescence. So if we have fluorescence on our y-axis here, and we're coming along cycles, we're detecting nothing, so we assume there's nothing there, it's just background. Then all of a sudden we're getting a signal. Okay? We're getting the signal with this one after 10 copies, all right? And then we get another one here after, we'll assume this one is 11 copies, I know it's not, but say it's 11 copies. Then the one that came up at 11 copies had less because it came up after it. But if it came up exactly one cycle after, it's got half as much. Okay? Because we know that every cycle there's a doubling. Hopefully that's reasonably clear. I'll try not to avoid the terms of logs and everything. But when we get right the way down to the end here, so fundamentally if we're looking at 35 cycles here, most polymerases and detection systems in real-time PCRs will, within a few cycles either way of 36, let's say, that'll be where a single copy can be detected. Okay, so that's a very, very small amount of DNA. We're not talking nanograms, or we're not talking picograms, we're not talking femtograms, we're probably talking attograms here, which is very, very small amounts of DNA. So small that it's hard to actually imagine. Okay, so just quickly going over again, this is a cyber-based assay. This is how I said this was originally designed. And the cyber is just a fluorescent dye that binds to the base pairing, so intercalates when those two strands are annealed. Okay? Only when they're annealed. And that's important for the latter case study. And that means that this signal during the annealing phase in the extension will be fluorescing. When it comes to denaturing time, when we go up to above 90 and they come apart, there's not going to be any fluorescence, okay, in theory, because there's no intercalation there. So that's important for a bit later on. So the advantages are, it is very specific. If it's designed well, there's no need to do anything else. Designed poorly, and there are issues. Why? Because we talked about very briefly in that original PCR that you could have aberrant products that aren't your particular sized band. If you have them happening also in your real time, they're also going to fluoresce. And you can't tell what is the band that you're after and what isn't. You don't see bands here. All you see is fluorescence. So if you have two products being amplified, you're getting signal from both of those. Okay, so there's a lot of optimization that goes into these to making sure that the cyber assay or the PCR is really only amplifying what you want it to. if you do that, then it's a beautifully specific, beautifully sensitive assay. Okay. And it's obvious that you can't multiplex because you try and put another target in there and you can't differentiate because they're both just going to fluoresce. Then we have our hydrolysis probes, and we're really meant to call them hydrolysis probes rather than TACMAN probes. TACMAN probe is a proprietary name, but hydrolysis means that basically when you have a probe which has a quencher and a fluorophore, okay, so here's our fluorophore, and here's our quencher, or up here like this. When this quencher is in close proximity, so when it's still bound to the probe, there's no fluorescence going on, it's quenching it. Okay? When the polymerase comes along during the extension step, the polymerase has a 5' exonuclease activity, and all that means is it's going to smash this probe up when it hits it. It smashes it up, and during that process, the quencher is separated away from the fluorophore. When that happens, it can no longer stop the fluorescence, and you're getting fluorescence happening. As soon as that happens, it's permanent fluorescence. Okay? It doesn't matter whether we're at denaturation temperature or annealing here. That fluorescence is going to stay. Okay? So you get this build-up of fluorescence with every cycle, because with every cycle you're going through this process of having the anneal, the probe sit on, the probe gets stripped off, and more fluorescence happening. So that's the fundamentals of the hydrolysis for TACMAN probe. Now, it also means that you're only going to get signal when this probe actually sits on what you expect it to sit on. Okay? So when your forward and reverse primers are specific and your probe is specific. So in theory, you've got another level of specificity in there. But if you've designed a cyber assay which doesn't have the probe, if you design the forward and reverse primers purely specifically as there, I mean, you can't get more specific than 100% specific, if you know what I mean. So the advantages with this are that you can actually multiplex. How can you multiplex? Because you can actually have different probes with different fluorophores that can give off different colours, let's say, different spectra, and that they can be visualized. So in one particular assay, in one particular sample, you can be looking for one target because the probe has, say, a fluorophore that is in one particular wavelength, and in that same sample you can be looking for something else because you're using a probe that can be detected on a different wavelength. Okay? And they don't cross over, or you hope they don't cross over. In reality, you do get these spectral crossovers, but that's a bit beyond today, I think. So once again, this would be the same deal. With increasing cycles, you're getting increasing signal. Okay, so if we're going to use this, then for an unknown sample to detect, we really need some sort of standard. You can use a PCR product as a standard, and you can quantify that PCR product and try and figure out how many copies of that you have, or you can refer to it in a weight. Likewise, you can do the same for genomic DNA, but there are always pros and cons with all of these. The PCR product, number one, you're going to be playing with lots and lots of copies. There's other things you've got to clean out of your reaction if you want to use it as a standard. It'll have had your primers in there. If it had other bands in there, you've got to get rid of those. So there's some issues with that. They don't necessarily last longer. They stick to the tube a little bit more readily than other things. So it might be fine on the day you make it up for a standard, but you might have to make it completely up from scratch the next time you wish to use it as a standard. Genomic DNA, useful, but it's very long, and it also has its issues. Plasmid DNA, so here we're talking about, we're taking some of the target or a sequence that has the target we're looking for, putting it into a vector, and then extracting it out. And we all know, hopefully we all know, that plasmids, or the common plasmids that we're using, or that you've been using, we've learned about are usually circular, okay, and that they have something called supercoiling. So supercoiling, you know what happens with your telephone cable, not that many people have landlines anymore, or let's say a rubber band. You can twist it up and it can knot into all sorts of formations. That can be problematic, okay? That can be problematic because if it takes on different conformations, if it intertwines, then it can be difficult for probes and primers to access it. So if we cut it, nick it, then it turns into a nice long linear strand, and then it behaves more like the target that we're trying to detect. So the fundamentals of this are, we want to use a standard that behaves as similarly to what we're trying to detect as possible. Okay? So that means that we really want it to be as close to usually genomic DNA as possible. What we're starting to use now is these synthetic gene blocks. They're, as molecular biology just shoots ahead, it's now quite easy to have a large chunk of DNA synthesized and very accurately quantified, and then we can basically have a very accurate amount of DNA. We know exactly how many copies and use that as a standard, so we can dilute that out, and so we can serial dilute that out, and then we can get beautiful standards happening.
That's just for measuring our length again.
No, that's for, say we have an amount, so we're basically putting, if we're trying to detect something, like a part of a gene, if we put part of that gene into that synthetic block, and then we dilute it out so that we set it off, so here we've got 1,000 or say 1,000,000 bits of the gene we're after. We can dilute it out, so the next one has 100,000. We have another sample that has 10 times less than that, so 10,000, 1,000, 10, 1. And we can then actually create a standard curve and see where our unknown hits, and then we can basically read off, okay, how many copies was in our unknown sample. So using that gene block. Okay. So it's just serial dilution. Here we've got an example of a dilution of some of our gene block. So our standard, say, here we go. We've got a very accurately delivered synthetic block of DNA that has 1,000,000 copies in it. Okay, and we can just dilute that out. So in every single tube or well of our machine, we have a known amount. We can then, on this axis, would be a quantification cycle. So for the moment, just ignore that this has a number on it, but this would be the cycle that the real-time detected on. Then if we had an unknown, and we say it had a cycle number of 20, then we just read along where 20 is, hit, come down and say, okay, this sample has 1,000 copies in it. Okay. This particular development has actually already gone through and converted the cycle number of the real-time into a copy number. But it's just showing the linearity over the whole dynamic range there. The limit of detection, the limit of quantification, they're measures of basically the sensitivity of your assay. Limit of detection is purely where you, there are lots of definitions by the way, but this is where we want to be. It's a measure of reliability of being able to detect. So we want to be able to detect with 95% confidence that, we want to be able to, with 95% confidence, detect what we're after. Okay, so if we've got, say, 10 wells, and we have 9 of those come up positive, 1 of them not, well, that's only 90%, so that hasn't reached our reliability level. So you can see the more replicates, if we had 100, we'd need 95 of them to come up positive. We can only afford 5 of them to come up negative for that to be a reliable detection method. Now, it's a bit beyond this course, but there's a a different distribution that comes into play when you're at really low numbers. The Poisson distribution, law of low numbers. So basically, if we dilute down to 1 copy down here, and we took a volume of that we thought was 1 copy, 37% of the time we wouldn't have anything in there. 37% of the time we'd have 1 copy in there. 18% of the time we'd have 2. So there's a problem there. The assay is really good at detecting a single copy, but if you've diluted down where you know that 37% of the time you're going to have nothing, then it causes noise down in this low area, so it looks like you're losing precision, but it's not really. The assay's not being less precise, it's just that this different distribution is coming into play. Okay, so that's why we have the linearity suddenly going a little bit off. It's also why we can't have limits of detection less than 3 copies. Okay? Through doubling, it's just an impossibility using a 95% ruling. If you to have a Poisson distribution saying that 37% of the time you're going to have nothing. Okay, so when you drop below 3 copies, it's impossible. Below 3 copies, you have somewhere around 30%, 37% of the time 0, and that's a far cry from the 95% required. And the limit of quantification is purely a measure of reliability for the ability to quantify. So in simple terms, it's the linearity of this line. Where does this line suddenly stop being linear? Once again, it seems to stop somewhere around the very low, in low numbers, because of that Poisson distribution coming into play. The good thing is, for Peruvians at least, with the amoeba, you can see that with approximately 3,000 copies in every cell, and you can't detect, where I'm really only interested in whole organisms, so 1 cell, 2 cell, etc. We're not really that interested in detecting kind of in this range where there might be some questions regarding sensitivity. Because if one organism's got 3,000 in it, it's not like we're needing to detect 1 copy. Putting that into use, we can also use those for swabs. The swab here is purely just taking like a cotton bud and wiping it over the gill surface, taking mucus. We then do a DNA extraction on that, and strangely enough, it seems to, well, it's not strange, it makes perfect sense that you get this with increasing pathology score. So the more obvious physical signs that there's been an infection with the amoeba, you get a lower cycle threshold. The lower cycle threshold means that the organism's been detected earlier, so there's more of it. Likewise, as you come down on our measure of pathology, where there's likely to be less amoeba, we end up with a higher cycle threshold, so less of our target. It's quite linear. So that's using it for swabs. So we've shown kind of the development of the assay. We've used it for some swabs. We also commonly use it for water. So we're trying to measure how many amoeba are in the water. So we filter a known volume of water through onto these glass fibre filters that obviously will trap the amoeba, and then we do a DNA extraction on these, and we try and get the cleanest DNA we can, and we put it into our assay. Now, what we found, and I can't really put the results up because some of them are confidential, I can probably see them even though they're being recorded, is that the amoeba numbers are really low, and we've used this throughout the whole world now. So this assay has been put to use by some of the big companies in Scotland, etc. Whilst the amoeba numbers are low, nonetheless they're there, and we know that it only takes 1 amoeba to cause a problem. So if you have 1 amoeba per litre, and you're going fish and farm fish there, you'll eventually have a problem, a disease problem from the amoeba. Okay. So I think it's pretty obvious there that we've shown some sensitivity. I didn't really show the specificity, but obviously those primers were shown that they only amplify that particular amoeba. And it's a relatively rapid assay. And I really hope it is, because I have probably 600 samples waiting for me to process in the next week. Okay, putting another assay into use, and this one becomes a little bit more complicated because we're starting to muck around with what you can and can't do with real-time detection. Okay, so once again, work we did here a few years ago now, published in this particular journal, so when was it, 2013, but now we're interested in detecting a pathogen, a blood fluke in tuna. So we'll just quickly go through this. We're probably all aware of the importance or the monetary value of tuna, especially in comparison to other fish species. It's a highly valued fish species, especially in the Asian market. We, in Australia, grow southern bluefin tuna, and then in the northern hemisphere, we're talking about Atlantic and Pacific bluefin tuna. Okay. Now, in Japan, they've actually closed the life cycle, and by closing the life cycle, it means they can just go right the way through. They don't have to take in wild tuna like we do in Australia. In Australia, we go out with a quota, grab with nets, catch juvenile tuna, tow them to South Australia, to Port Lincoln, and then sit there throwing bait fish at them to grow them up, to fatten them up. In Japan, they've successfully closed the life cycle, so it's more like a traditional farming practice as you'd see done for, say, Atlantic salmon, if you're more familiar with those. However, whilst we don't have too much of a problem with our ranch, we call it, where we've gone out and collected the tuna from the wild, the ones that are cultured in Japan do have a problem. They seem to be very susceptible and break with these blood flukes, which do cause disease. Now, whilst there are blood flukes in the tuna here in Australia, and there maybe some people may think that they're associated with some of the issues, they don't really have a traditional disease problem in Australia. However, in say Japan, the young juvenile fish are can be heavily infected and will obviously die. There are some different species here. This is important to know. So there's forsteri, Cardicola forsteri is the blood fluke that's detected in our southern bluefin tuna in Australia. It's also in Atlantic bluefin tuna. But in Japan, these are the two that are kind of the most problematic, Orientalis and Opisthochus. Now, when we started doing this work, it was only thought that forsteri was the only fluke present here in Australia, but we soon found differently. Okay, so the life cycle, a little bit important for what I'm going to talk about. It's because it's a cycle, it's a bit hard to figure out where to start from, but say we start from the host that's infected, they release Myricidae, which then go to another host. This is talking about a polychaete, so this is down on the benthos. It will then release sporocysts, which turn into cercaria, which go through the water column, back up, enter the fish, and make their way to the heart. The adults in the heart here release eggs. The eggs go up, get trapped in the gills. So there's the life cycle. Traditional methods, we use parasitology, quite invasive obviously. You have to rip out an adult's heart and have a look for flukes in there. These are very valuable fish, so they'd rather not do that. Or histopathology, where you're actually once again working with basically a necropsy. So to use molecular approaches, we've been able to basically develop a non-lethal, so we don't have to kill our fish. They're very valuable. Farmers don't really want you to kill them, so we can take blood and we can actually measure the amount of pathogen DNA in the plasma component of the blood. Okay. Now, interestingly here, there's no actual eggs or pathogen as such, adult there. We're talking about free DNA. That doesn't necessarily mean that there's a live pathogen there. It just means that there has been a live pathogen there. However, if we filter the water and use that method to detect the pathogen, we really are only going to be detecting these two life stages, and we can't differentiate between those two. So, nonetheless, the assay that we developed is sensitive enough to detect those. We've gone through the cyber green assay, we've gone through the hydrolysis probe assay before. The method that we used in this particular study was a bit of a combination of both. Okay. This is an actual hydrolysis probe that we're looking at here, but it's a common reporter one. So fundamentally we have two primers, but on the end of one of the primers we have a section that allows an area for the probe to sit. It's almost a bit of a cheats method of the traditional hydrolysis probe or TACMAN probe, but nonetheless it still gives you the specificity in that you won't be getting signal derived from other bands or primer dimers, etc. However, there's still no melt curve analysis as such for that. This is a little bit extraneous. You don't really need to worry too much about all the different primers and probes. Specificity testing. You could probably go through this at your own leisure when it's on Milo, but fundamentally it's just telling you that you only get the signal or the band when you put the appropriate matched primers and pathogen in there. Likewise for the common reporter TACMAN. Interesting results come about when you start to use duplex. So this is where I said you can actually have your hydrolysis probe with different fluorophores on there. We can detect the two, which is useful. We can not only detect, but we can actually quantify with that. And we once again are able to, using those terms, the limit of detection and quantification, which I explained for the previous case study, to show you that the limit of detection is somewhere around 5 or 2. I could ask the tricky question, and is it really possible to have a limit of detection of 2 for Forster I? No, it's not, because if you remember what I said before, when you do get down to less than 3, if you've got your limit of detection set at 95%, the Poisson distributions, the law of low numbers states that you can have 37% of the time with nothing. Okay, so either there's been a slight miscalculation in the standard, which has allowed that, or we haven't had enough samples to actually have a true Poisson distribution, and just by chance we've been able to have 2 appear. I've said, and we'll end pretty quickly here, Chris was right, my verbal diarrhea did come to the front. I said that the cyber and the hydrolysis probes, if the cyber is designed really well, is very specific, and there's no real advantage to having a hydrolysis probe in terms of specificity and sensitivity. There is a particular case where it does come into its own, and that is if you have a lot of other DNA surrounding or in your assay. How would that come about? Well, we're not really going to be looking for something in amongst lots of pathogen DNA, but if we're looking for a pathogen DNA in the host, and we've somehow co-extracted a lot of the host's DNA, so in this case a lot of tuna DNA, if we're using the cyber assay, there's the chance that we're going to have that DNA fluorescing, which means that it's going to mask, fundamentally put the background, the baseline really high, and it's going to mask a very small amount of the actual pathogen. That doesn't occur with the hydrolysis probes because that background DNA isn't actually generating any fluorescence. So that's the real beauty of the hydrolysis probes. Putting these into use, we're able to show that the PCR approach in terms of positive percent agreement and negative percent agreement was a superior assay. Fundamentally, this is just showing you how good an assay is compared to traditional assays. So it's the PCR based versus the traditional heart flushes, etc. Interesting thing was, put that into practice, and we actually showed when we used the assay to detect the 3 different cardiacola, so blood fluke species, we found that, okay, in southern bluefin tuna here in Australia, it seems that not only are there more than one species, Forster is definitely not the only blood fluke species, but it's not even the most predominant species, Orientalis, which is here represented by the green fins. So that was a very interesting finding with some great implications. Likewise, when we look in water, this is just to show that it's been used in water, we can see that it's actually the most prevalent in the water compared to Forsteri as well, with the pluses on the far right. Here's just a little column showing the distribution from a certain amount of cages. What does this mean? Other than the summary here, just reiterating what I've spoken about. It's played out quite interestingly. This was a few years ago. They do treat the tuna here in Australia with, or have trialled some drugs. And interestingly, they started trialling a particular drug, praziquantel, and what happened? We've no longer been able to detect Orientalis, but we can still detect Forsteri. So there are some definite managerial kind of implications here. So it looks like praziquantel at the moment is specific for one of those particular blood flukes. Okay, and I'm done. Thank you very much for listening.
Questions?
No, I rushed over kind of technical terms there quickly, but it was hopefully an overview of how they can be used.
In terms of costs, is it more expensive to do something with a hydrolysis probe compared to undecided cleaners?
Well, yes and no. What the development of the hydrolysis probes do cost more to manufacture, but once they're up and developed and you have them, then it doesn't cost any more to actually do your run.
So it's a startup cost difference, I guess. You've got some money up front for the manufacturing of the probe, but then the costs are pretty similar, aren't they?
Yeah, I mean, 15 years ago when I started first using hydrolysis probes, they were hideously expensive. Now you can get them manufactured for a tenth, if not less, of the price to back then. So cost is not really an issue if, well, it obviously depends on the value of the result. It's going to save you thousands to have a correct answer. The good thing is, you, as a scientist, don't do it yourself. These are manufactured, so by the people who synthesize DNA, the primers, etc. So, I mean, you can do it yourself, but it's not that difficult. It's just a matter of mixing things together. The difficulty comes in actually assessing the purity. So, being able to show that 99.9% of your probe is actually attached to the primer.
So what do you think's driving the difference between the cardiacolas response to praziquantel?
I don't know, and I probably shouldn't have even spoken about it. But no, it's a very interesting
It's an
finding.
academic question, you know. You wouldn't necessarily
but also it wasn't expected, like from the majority of blood flukes are very host specific. So to actually have 2 in there, and it's not only here but also in Japan as well, is quite interesting finding in its own right. To detect them in the water is also a very interesting one, because you've got those life stages, so the Myricidia and the Cercaria. So we've actually been able to use this assay to show that this is also very recent work that I can't put up figures for, but we can pretty much show that the Cercaria, which are released from the polychaete intermediate host, they release the Cercaria. Those Cercaria then seem to enter the host through the gills, and then once they're in the gills, make their way down to the heart.
Any more questions, or should we let Andrew off to go back to the bench? I think he's
The really
currently extracting hundreds of tubes of samples.
Yeah, that's right. I've already done my little whinge. Right. Yes. So I forget the portion of the Japanese domestic market that is actually that comes from domestic tuna market that use aquaculture tuna, but it's growing. It's a significant part now. A few years ago, it was more, well, look, we can close the life cycle and we can grow them. We have a few disease issues, but now we can produce enough of them to actually start to be a proper player in the tuna market. They treat heavily with praziquantel, heavily. But then again, they also put formalin on their sashimi and the like. Horses for courses.
Okay, no more questions for Andrew?
All right, thank you.
Thanks, Andrew. Carries his own seating.